Hi, this is Peyton, and along with Susie, Priscilla, Rita, and Ava, we did our topic on gene therapy. So first, we'll begin with an introduction to the topic of gene therapy. So this is the technique of altering an individual's genetic material for clinical benefit. So if a person has, say, a disease or cancer that can be linked back to a mutated gene, you can correct this in cells by introducing external genetic material. And this genetic material is typically delivered by vectors like viruses. Viruses are already excellent vectors because they have evolved the ability to invade and replicate in cells. And of the viruses, the most extensively used are retroviruses, adenoviruses, and parvoviruses, which each have their pros and cons. So retroviruses are small, they don't have as many genes, but they can integrate into the host genome so they can last effectively forever. While adenoviruses have a larger genome and they can have like a lot more genes and possible proteins encoded. Parvoviruses, again, are small, but they can evade host immune defenses a lot better. So there's quite a range. And I'll talk about two of the three types of gene therapy, the first one being gene augmentation. So this is for treating loss of function disorders like cystic fibrosis, where you introduce a functional copy of a gene that has lost its original ability. So this is really useful in treating viruses, using viruses to treat immunodeficient people where you can use retroviruses. These are usually the most effective because they can integrate and they'll be effective permanently. Another type is gene inhibition. So this is where you have a gene that has a faulty activity. So usually it's overactive or has unusual activity, and you'll interfere with this to block it by either blocking the direct expression of it or blocking the product produced by it. And then this is useful for cancer treatment, where you're going to modify viruses to target tumor cells and then either block their activity or kill them.
All right, and then the third type of gene therapy is the killing of specific cells. And this is typically used for cancer treatment, where the elimination of a specific subset of cells aids in relieving and curing the disease. And cells can be targeted one of two ways, either through the insertion of a gene which directly causes cell suicide, or through the insertion of a gene which, when expressed, produces a protein that marks the cell to be killed by the patient's own immune system. And cancer cells are very easy to target for this type of gene therapy because they possess many distinct qualities which distinguish them from healthy and normally dividing cells, including abnormal mitosis, abnormal extracellular protease function, and increased susceptibility to infection and disease. Right, next. Right now, almost all gene therapies are still in the clinical trial stages, although the US FDA has approved about 24 for sale in the US. And therapies can be used to target either somatic cells, which are non-sex cells, where any genetic alterations affect only the patient and aren't inheritable, or germline cells, which are sex cells, in which case the alterations are inheritable to all future generations. Gene therapy has a wide range of potential applications, including use in regenerative neurosurgery, which would allow for treatment of strokes, traumatic brain injuries, and diseases such as Alzheimer's. Right now, several clinical trials are also underway for the treatment of Parkinson's with promising early results. And the vast majority of gene therapy clinical trials right now are for the treatment of cancer, it's about 66%. All right, next. There are also a lot of challenges to gene therapy because of the use of a viral vector being used to introduce new DNA. The viral vector may trigger a severe immune response, which can lead to organ failure or death. The vector may impact cells outside of the intended target, which could cause cell death and injury and lead to new diseases such as cancer. And while genes that cause virulence in viruses are removed prior to being used by as a vector, there is always a chance that the virus could regain the ability to cause disease. If the vector fails to insert the new DNA in the right location, this can also cause cancer in the patient. So going forward, gene therapies need to be developed with increasingly reliable methods of introducing the new DNA and new ways to eliminate the severity of side effects. And right now there's a slight controversy regarding the use of germline gene therapies, and it's currently banned in the USA.
So now we're going to move on to our paper, which is gene editing and elimination of latent herpes simplex virus in vivo. Next. So the main goal of this paper and this study was to pretty much improve on previous studies that used CRISPR-Cas9 delivered by AAV and to provide a new therapy because the current therapies for herpes pretty much just leave acute symptoms. Previous studies using Cas9 delivered by adeno-associated viruses resulted in very low levels of gene editing. So the researchers of this study wanted to improve on that by replacing CRISPR-Cas9 with meganucleases. They simultaneously targeted different areas within the HSV genome, and they added more meganucleases relative to the HSV infected cells. So with that, they hypothesized that using meganucleases instead of Cas9 and improving distribution and compatibility of AAV vectors would result in an increase in HSV editing. Next.
The virus being examined is herpes simplex virus, or HSV1, chosen primarily for its ability to go latent within the neuron. There are 3 meganucleases used in this experiment which recognize certain sequences within the HSV genome, and these are M5, M8, and M4. To deliver these meganucleases, adeno-associated viruses were used, and the 3 serotypes were AAV1, AAV5, and AAVrh10. The bacterium Staphylococcus aureus was also used for CRISPR and Cas9. Next.
So the CRISPR-Cas9 gene editing system was tested first in this study, and the basic procedure for this was to inject mice with live HSV and allow 28 days for latent infection to be established. And at this point, then the 3 treatments were administered to different groups of mice, with the first being a control in which nothing else was added, the second being an adeno-associated viral vector, which was carrying the Cas9 system, which had 2 short guide RNAs to target 2 different sites within the HSV genome, and the third treatment being the same vector with an encoded single meganuclease to target 1 site in the HSV genome. Next. And these are the results. Figures B and C are showing that a high level of latent HSV infection was established to a similar degree in all the test groups, which shows that the injection was successful. Figure B is showing the superior cervical ganglia cells, which is what SCG stands for, and TG in figure C stands for trigeminal ganglia. And then figures D and E are showing the levels of gene editing that was accomplished, which is measured by the % of mutation that was detected by next-gen sequencing. And you can see that at both sites of the short guide RNAs in the superior cervical ganglia, there was almost no detectable editing. And in the trigeminal ganglia, there was a very low percentage. In both graphs, you can see that the gene editing accomplished by the single meganuclease was much higher than the dual Cas9 system. Next.
This diagram provides a short summary of the different serotypes used in this experiment. Mice were injected with either 1 serotype, dual serotypes, or triple serotypes. The superior cervical ganglia and trigeminal ganglia were analyzed for HSV load with droplet digital PCR. Next. Analysis of the superior cervical ganglia and trigeminal ganglia showed that all of the adeno-associated virus combinations, except for AAV1 alone, transduced the ganglia at similar levels. The y-axis is the AAV genomes for every 10 to the 6 cells in the ganglia. The x-axis represents the serotypes used. Next. Dual meganuclease therapy delivered using AAVrh10 either alone or in combination with 1 or 2 of the other AAV serotypes led to the highest loss of latent viral genomes from the superior cervical ganglia and trigeminal ganglia. The greatest decrease in viral load was obtained in the triple AAV combination. The y-axis is HSV genomes for every 10 to the 6 cells in the ganglia. Next. These graphs analyze gene editing within the M5 targeting meganuclease. Gene editing was quantified by a T7 endonuclease 1 assay in which T7 endonuclease 1 recognizes and cleaves non-perfectly matched DNA. The y-axis represents the percentage of mutation by T7. Despite the efficient elimination of the HSV genomes, gene editing in the residual HSV genomes was low, generally less than 10%. Next. These graphs analyze gene editing within the M8 targeting meganuclease. In the HSV M8 site, gene editing was even lower and ranged from undetectable to only around 8%. There's even less concentration of gene editing in the trigeminal ganglia. Next.
So after they found out that Cas9 wasn't effective and that the Rh10 serotype for AAV was the most effective at delivery, they compared the effectiveness of a single meganuclease treatment versus a dual meganuclease treatment. So the single is M5 or M8, whereas dual includes M5 and M8. And in figure B, it shows that the dual meganuclease treatment resulted in a significant loss of HSV genome load in infected cells compared to single meganuclease treatment, which resulted in no significant loss of HSV genomes. It just resulted in moderate amounts of HSV genome editing. To further support this, they did another test with the M4 meganuclease, which resulted in a significant loss of HSV genomes. And this is mainly because M5 and M8 together and M4 alone, they can make double-stranded breaks within the DNA, which is why they can cause a significant loss in actual genome load compared to the single meganuclease treatment, which just creates single breaks, which can be easily fixed within the DNA. Next. So after this, they took TG and SCG samples after the dual treatment, and they found significant loss in HSV genomes. This is just to further support the idea that the dual meganuclease treatment is much more effective than the single meganuclease treatment. Next.
The results of this study show that the optimization procedures used were effective, and that with these optimized procedures, it is possible to reduce levels of latent HSV infection by significant amounts. The best that was achieved in this study was a 90% decrease in HSV genomes in the superior cervical ganglia and a 50% decrease in the trigeminal ganglia, which was accomplished using the dual meganuclease to target 2 sites within the HSV genome and multiple serotypes of the adeno-associated vector, which targeted different subsets of cells at different rates. This reduction is high enough to show that it could be a possibility in the future for this type of gene therapy to be effective in humans. And the results have been somewhat replicated in other studies, which have shown that similar viral vector gene therapies can reduce latent infection in mice infected with HBV or HIV. And all of this together shows that in the future, this kind of gene therapy could potentially represent a cure for latent viral infections. And these are our sources. Thank you.